Our protocol demonstrates that the ECM may promote adipogenic differentiation in the absence of classic adipogenic mediators, and is the first to demonstrate disease-specific regulation of cellular metabolism by the ECM. Our technique provides a tool for dissecting the roles of the ECM and adipocytes in adipose tissue metabolism and permits study of the role of the ECM in regulating adipose tissue homeostasis. Compared with standard 2D cell culture, our 3D model allows for more robust examination and dissection of crosstalk between adipocytes and adipose tissue ECM in the context of type 2 diabetes. We recommend beginning with smaller samples to gauge how well they decellularize. Keep in mind, there is interpatient variability in how well the cells grow and how well the tissue decellularizes. It is important to observe the tissue before and after each step to ensure the tissue is becoming decellularized and to understand potential differences between samples.
After obtaining visceral adipose tissue, or VAT, from surgery, prepare it for storage by adding 5 to 10 grams of the tissue to 15 to 25 milliliters of freezing buffer solution in a 50 milliliter conical tube. Completely immerse the sample and store it at -80 degrees Celsius until decellularization. For pre-adipocyte isolation, place 2 grams of a fresh sample of intact VAT in 20 milliliters of collagenase type 2 solution. Then insert sterile scissors into the tube and thoroughly mince the tissue.